what projects you're using our technology with.
Most important aspect is the measurement of motile responses in metastatic cancer cells. There are two important aspects we follow. This is focused on responses of cells derived from biopsy from patients, which we can analyze within a few days from being taken out of the patient, and see the responses to drugs which the oncologist would consider. And then using not only the cells from the patients but also established cell lines for testing of potential drugs. So this is drug discovery project.
Can you review for us why it's so important to understand this link between motility and cancer metastasis?
The motility is the crucial key to the metastatic cascade. Whatever genetic changes the cells may undergo or epigenetic changes in protein expression, these changes always result in active cell motility. Without that, the metastasis cannot form. So I believe that this is the critical aspect, and if we can target that, whatever the genes are involved, we'll be winning with the fight against cancer.
Do you combine quantitative phase imaging with other imaging techniques?
Yes, we combine it especially with fluorescence, which is very beneficial in providing information about distribution of specific molecular species, whereas the Q phase gives us the information about the cell. So the Q phase is telling us about the motility, about the dynamic morphology of the cell, and the fluorescent complements it with the individual specific proteins which can be labeled and visualize in context.
So how did Q phase help enhance your work?
It's mainly in its highly quantitative aspects, where we can see the heterogeneity of the cells, can exactly measure the motile responses to treatments, whether it's genetic or drugs.
What is this video showing that you took using Q phase?
This is a playback from a time-lapse recording with HT1080 human fibrosarcoma cells in culture. We can see individual cells moving around, and some of them are dividing. The color coding is calibrated in picograms per micrometer square. So the hot red colors is the highest density of dry material. The scale bar at the right bottom here represents 100 micrometers. So the whole field is about 600 micrometers, so it's like half a millimeter wide. In the left-hand side graph, we see the growth curves. So it's the integrated mass in individual cells as they are growing, increasing in mass in time, and from this we can derive how rapidly they grow, even in shorter period of time, although here we recorded them for 25 hours. The right-hand side graph shows detailed tracks which we took 150 minutes intervals, and these short tracks we moved to a common origin, and we can see the majority cells don't go that far from the center, and a fraction of them achieves the highest translocation. The tracks end with the black dot.
What kind of researchers would you recommend the Q phase to?
To any researcher aiming for measuring behavior of cells in culture, where they would be interested in the heterogeneity of cells, intact cells, without labeling and without significant phototoxicity. The cells can be of any origin. We also experimented, for example, with cells derived from plants.